It is called as the octant stage or the 8-cell stage of the proembryo. This proembryo now undergoes a periclinal division. Before this lockdown, if you remember, I have explained, especially to the 11th or 12th O section, I have already explained the various planes of division, periclinal plane and the anticlinal plane. Periclinal plane is always parallel to the outer surface. So now these 4 cells on this plane and the next plane, they are going to divide in this manner to form 8 cells and then 8 cells. So total number of cells now will be 16 cells. Now up to this 16 cell stage, it is referred to as a proembryo. Now things are going to change. The outermost cells which are 4 in this tier and 4 in the below tier are going to divide and develop into the epidermis. The inner cells are going to divide and develop into ground meristem. Remember the 11th class topic, ground meristem is supposed to give rise to pith in the cortex and will give rise to procambium, which will give rise to the pericycle, xylem, phloem. So that is also referred to as stele. So I'm repeating again. The first cell situation was the zygote. Zygote underwent growth. After growth, it developed a cell wall. Then underwent an unequal cytoplasmic division transverse plane, which gave rise to a basal suspensor cell and the tip embryo. Now this embryo and the basal cell underwent a transverse and a vertical plane division to form a 4-cell situation called as tetrad. Here the proembryo carries only 2 cells. The hostorium developed from the tip cell, and this cell will undergo 3-4 times, 5-6 times divisions to form a long chain of cells which are 7 to 8 cells. This whole structure is called as suspensor. The 2-cell stage of the proembryo underwent another vertical plane division and will cause the formation of 4-cell stage. 4-cell stage underwent a transverse division to form 8-cell stage. 8-cell stage proembryo undergoes a periclinal plane of division to form 8 plus 8, 16-cell stage. The outer cells will develop into epidermis. The inner cells will develop pith and the cortex, as well as the xylem and phloem and pericycle, that means stele. Once all these divisions are done, it will develop into a globular embryo. The cell of the suspensor which is immediate to the globular embryo is called as hypophysis. Hypophysis undergoes division in longitudinal manner as well as the transverse manner to form 3 layers or 3 tiers of the cell. Now these are the 3 tiers of the cell. The outermost one will give rise to the root cap initial. The next one will give rise to the protoderm or the root epidermis, and the innermost one will give rise to cortical tissue of the root. Now this whole thing will be the suspensor. So please remember, embryo has some contribution even done by the suspensor, and only the last cell of suspensor, and that suspensor cell is called as hypophysis. By this time, this embryo has started developing into a heart-shaped structure because cotyledons are coming up. This will develop into the shoot apex, and finally this develops into a shrinking type of suspensor because the food requirement is finishing. Then root cap will develop from the hypophysis, then radicle will develop from the hypophysis. This will be the embryonal axis. This is the epicotyle and the plumule, and these are the 2 cotyledons. Now this is the way a dicot embryogenesis takes place. A dicot embryogenesis. There are certain differences in dicot and monocot embryogenesis. Now what exactly are those differences? So monocot only carries 1 cotyledon. So the other cotyledon is not going to develop. It will remain as a shrunk portion but still visible, and it is called as the epiblast. In the case of grasses, such a cotyledon is also called as scutellum. It is highly reduced. Reason is abundant amount of food is still present in the endosperm. Please remember one thing, that endosperm is required during embryo formation as a food. In dicot plants, the food from endosperm shifts slowly into the cotyledons. So cotyledons start swelling and enlarging in size. So they are storing the food for the seed till the point of germination. But in the case of monocot plants, the cotyledon do not grow big in size, so they don't store a good amount of food. So food remains in the endosperm. So monocot seeds will always have endosperm present inside them even at maturity. But the dicot seeds generally do not carry the endosperm in the mature stage. Reason is cotyledons. All the food has been shifted to cotyledons, and cotyledons provide the food during the seed germination. So this is the main difference between these two. You can check one more thing. The embryo of the monocot also carry a coleorrhiza. Coleorrhiza is a undifferentiated sheath which is protective in nature